Welcome to the photosynthesis lecture in Bio 120. Let's take a look at how microorganisms perform photosynthetic reactions. Photosynthesis is defined as a generation of ATP and NADPH by light-driven reactions where carbon dioxide is fixed into cell carbohydrates. So microorganisms that perform photosynthesis can be divided into oxygenic phototrophs and anoxygenic phototrophs. Oxygenic phototrophs are mainly cyanobacteria. Anoxygenic phototrophs can be divided into 6 major phyla: the proteobacteria, including alpha, beta, and gamma proteobacteria; the Chlorobi, which includes the green sulfur bacteria; Chloroplast, which are the green non-sulfur bacteria; the gram-positive firmicutes and acetobacteria; and the gemmatimonids. This phylogenetic tree shows you the relationship with these microorganisms, and as you can say from the phylogenetic tree, the capacity to perform photosynthesis doesn't fall within exclusively only one group of bacteria. So phototrophs can be subdivided further into photoautotrophs and photoheterotrophs. Microorganisms that fix carbon dioxide are the photoautotrophs, and microorganisms that use organic carbon as a carbon source are the photoheterotrophs. Examples of heterotrophs include the purple non-sulfur bacteria, the green non-sulfur bacteria, and heliobacteria. Microorganisms that use oxygenic photosynthesis are cyanobacteria. Cyanobacteria derive their reducing power from electrons that are given from water. The energy is derived from light, and those electrons and ATP energy is eventually used to fix CO2. Now, purple and green bacteria are able to perform anoxygenic photosynthesis. Another molecule other than water will donate the electrons, for example, hydrogen sulfide, and those electrons eventually will be used to fix CO2, as light as well is used to produce ATP. So let's take a look at the photosynthetic pigments that are used in bacteria. Most of the studies that we have seen in Bio 110 were looking at the chloroplast, and both chloroplast and cyanobacteria use chlorophyll A, shown here on the left. Bacteria which are photosynthetic use a different molecule called bacterial chlorophyll A. Here in the right, you can appreciate the absorption spectra of bacterial chlorophyll A as well as chlorophyll A. Chlorophyll A is shown from the alga Chlamydomonas in green, and the purple bacterium Rhodopseudomonas palustris is shown as the microorganism containing bacterial chlorophyll A. As you can appreciate from the graph, the absorption spectra of the bacterial chlorophyll A and chlorophyll A is quite different. So chlorophyll, as I mentioned, is found in major plants, algae, as well as cyanobacteria. All other photosynthetic microorganisms have bacterial chlorophyll. Figure 13.3 shows you the different kinds of bacterial chlorophyll available for microorganisms that perform photosynthesis. What you can see in the right hand side, it's a skeletal image of the bacterial chlorophyll, including all the 7 R groups that makes it different across all bacterial chlorophyll molecules. When you look at this table, look at the color that it matches the R groups. For example, R1 in bacterial chlorophyll, it's represented by this group right here. And as you can appreciate, most microorganisms like bacterial chlorophyll A and B have the same motif, but that changes for bacterial chlorophyll C, which now has a hydroxyl group and a methyl group. So what you can appreciate is the different types of chlorophyll that are present in microorganisms. The other thing that I want to point out to you, it's the microorganism that contains that chlorophyll. So for example, bacterial chlorophyll A, it's here saying that it's found in purple bacteria, and it also gives you the absorption maxima that the bacterial chlorophyll has in vivo. So you can also appreciate that the different bacterial chlorophylls have very different absorption spectra. Now, as you have learned in Bio 110, the chloroplast is the organelle that harvests the energy of light to generate ATP for plants to fix CO2. It is found in algae and plants, and it basically looks like a cyanobacteria. So figure 14.4, shown on the top, is showing the thylakoid membranes within a cyanobacteria. You can appreciate the dense array of membranes inside the cyanobacteria that strongly resemble the structure of the chloroplast. And as you remember from Bio 110, this is a very organized structure. The chloroplast is a triple membrane structure containing an outer membrane. The outer membrane is containing in the inside an inner membrane. And the inner membrane has membranes that we call the thylakoid membranes. Thylakoid membranes will stack to form grana. Here in the thylakoid, you have a separate environment inside the thylakoid, as well as the outside of the thylakoid. The outside of the thylakoid is called the stroma, which is quote-unquote the cytoplasmic region of the chloroplast. And as you remember, the reactions that are happening inside the chloroplast stay within the cytochloroplast. So the ATP as well as NADPH that is made in the chloroplast doesn't leave to be used by the cell. Now, when we look at the capacity of microorganisms to harvest light, what we're going to see is that they contain structures that allow them to collect the light in which they're shining. So in this particular image, what we have is the reaction center that contains the chlorophylls or bacterial chlorophylls required to convert the energy from light into electrons to initiate an electron transport chain. Here in the reaction center is where you have the electron transfer. Now, what you also have is a set of light harvested antenna, here shown as LH1 and LH2. And these light harvesting antenna are an array of bacterial chloroplasts or chloroplasts, depending on what you're looking at, that are able to harvest light and funnel that energy to the reaction center in the form of resonance. And there is no electron transfer when the energy is passed as resonance. So what you have in the right here, it's the atomic force microscope structure of a reaction center and also the light harvesting center that are surrounding it. As you see, it's a very organized structure that is able to harness the majority of light in which the microorganisms are exposed and bring that energy to the reaction center to initiate the electron transport chain. Now, some microorganisms have very specialized structures that allow them to harvest light. Let me introduce you to the chlorosomes in green sulfur bacteria. An example of this can be found in Chlorobium tepidum, which is a green sulfur bacterium and an aquatic microorganism. Now, the chlorosomes is a specialized structure. You can see over here in this electron micrograph as the sphere where the arrows are showing. And when you look at one of those structures, what you have is an array of bacterial chlorophylls. So you have antenna bacterial chlorophylls of the C, D, or E type, which are arrayed in a crystallized structure. To that chlorosome, you have certain proteins which are called the FMO. These FMO proteins are shown over here, and they stand for the Fenner-Matthews-Olson proteins, which also contain bacterial chlorophyll A, and they're able to help transfer the energy harnessed by the bacterial chlorophyll in the chlorosomes directly into the reaction centers shown here in brown. Now, the chlorosomes, it is maintained together by a set of base plate proteins that attach the chlorosome into the membrane, shown over here in brown. What you also have is that because of this exquisite array of bacterial chlorophylls, the Chlorobium tepidum is able to grow at the lowest light intensities of all phototrophs. So it's a microorganism that could be close to the bottom of a pond, for example, and harvest the little amount of light that comes down by having this crystal array of bacterial chlorophylls that allow them to expand and harvest light. Other pigments are also important in the photosynthetic reactions, not always to help harness light, but also to eliminate reactive species that are generated by light. So let me introduce you first to the carotenoids. What we have over here in this image from figure 1412 are different purple bacteria with different mutations in the carotenoids. As you see here in this image, Rhodospirillum rubrum is a reddish microorganism. Next to that, you have a mutated version of Rhodospirillum rubrum in which the carotenoids have been removed. And as you can appreciate, the bacteria went from having a reddish color to now having a cyan-looking blue color. Rhodopseudomonas globiformis are also red, and they have a different kind of carotenoids than Rhodospirillum rubrum. Now, Rhodobacter spheroides, shown over here in an orange color, and here to the right you have a different strain of Rhodobacter spheroides that is missing the carotenoids, and therefore now it's brighter yellow. So what are carotenoids? Carotenoids are hydrocarbons that contain alternating single bond or double bond arrangements. They are characterized, for example, by beta carotene, shown here at the top in figure 13.8. Now, they are extremely hydrophobic molecules, and therefore they like to stay close to the membrane. They absorb lights on the blue region of the spectrum, which is one of the highest energy lights that microorganisms will be exposed to. And they're responsible to giving a lot of the colors, as I showed you earlier in the previous image, to those anoxygenic microorganisms. Now, they remain closely associated with either the chlorophyll or the bacterial chlorophyll in the photosystemic pigment complexes. However, those molecules do not work to generate energy. What they're doing is that they're able to take those toxic form of oxygen, in particular the single oxygen, which are formed by light. And therefore, the carotenoids quench those toxic oxygen species, absorbing a lot of the harmful light that will be able to produce them during photosynthesis. Now, another type of molecule that is involved in the light harvesting mechanism are the phycobiliproteins. What we're going to look here in this image is that phycobiliproteins are proteins that can absorb light. They form a structure called the phycobilisome, and the phycobilisomes are found, as shown over here, in the light harvesting systems of cyanobacteria and red algae. They are not found in anoxygenic microorganisms. They are attached in the thylakoid membrane. So here in this bacteria that you can see here, to the right, this transmitted electron micrograph, you can appreciate those spheres that are present in the internal membrane, and those are the phycobilisomes. As shown in the middle image, the phycobilisomes are usually attached to the membrane close to the reaction centers, especially the reaction photosystem 2. They are composed of multiple units. They have a tetrapyrrole ring, which are called billings, and those billings are able to absorb light, and they are coupled to proteins. So if you can see, imagine a chlorophyll molecule open up and now linearize, and that is basically the structure of a phycobilin. Now, the phycobilisome contains proteins as well as the billing molecules. Some of these proteins are going to be, for example, phycocyanin, which is a protein that absorbs light at 620 nanometers, as well as complexes of allophycocyanin, shown here as AP. Now, the bilin phycocyanin structures are going to be organized together, radiating around the allophycocyanin structure, and the agophylin structure is going to be close together to the second photosystem. Now, as light energy is harvested by the tetrapyrrole rings, they can then pass that light and resonance to the phycoproteins. In this case, for example, phycocyanin. Phycocyanin, having an absorbance of 620, can later pass that energy in form of resonance to allophycocyanin, which is a much larger absorption spectra at 650. Once the energy is harnessed by allophycocyanin, it can then be passed to the photosystem 2, which has an absorber maximum of 680. So in that regard, what the molecules we think the phycobilisome are doing. It's helping collect light of different wavelengths to bring it to the reaction center, enhancing the reaction center capacity to start an electron transport chain. So let's look at the spectra in cyanobacteria. And here in the big graph to the right, we have the chlorophyll A peaks. So as you can appreciate, there is a peak close to 430. There is a peak that is also present around 700, and the phycocyan is able to also give a peak close to 620. Now, look here in the small picture in the right where the arrow is present. That peak generated by phycocyan is missing here. So the presence of phycocyan is able to broaden the wavelengths that is able to be harvested by the microorganism. Most of the biology of photosynthesis that you have learned has been in eukaryotes. What this next section is going to do is to take a look at anoxygenic photosynthesis carried out by bacteria. So let me give you an example over here. This is the surface area of water in figure 14.9, and you can appreciate a beautiful color of purple that it's really not melted bubblegum. That is actually colonies in the biofilm of Chromatium oceni, which is a purple bacteria, which is photosynthetic. Now, when you take a look at the internal structures of those photosynthetic purple bacteria, you can find that microorganisms have extensive membrane arrays called lamellae that are going to allow them to harness energy. Here, Ectothiorhodopspira mobilis is one of the organisms that is showing the lamellae. But they also could have membrane structures formed in spheres in the chromatophores. So Allochromatium venosum, shown at the bottom here, has the chromatophores in beautiful little circular structures inside the cytoplasm. Now, both the lamella and the chromatophores are continuations and invagination of the cytoplasmic membrane and contain the proteins that are going to be involved in the electron transport chain of photosynthesis. Let's take a look at the reaction center of the purple photosynthetic bacteria. So as we look in the previous image, those purple bacteria have extensive membrane systems inside their cytoplasm that are used for photosynthesis. And in this particular case, the reaction center contains three major proteins: the L protein, the M protein, and the H protein. Within the H protein, you're going to contain the special pair, and the special pair are bacterial chlorophylls that are able to absorb light and initiate the electron transport chain. What you have here in this image from figure 13.11a, it's the bacterial chlorophylls that are part of the reaction center. The first two molecules that I would like to introduce you are the bacterial chlorophyll A, shown in red. Those are the ones which are going to initiate the electrons transport chain. They're able to then pass electrons to another set of bacterial chlorophyll A molecules shown here in yellow. Eventually, electrons from those bacterial chlorophyll A's will go to a third type of molecule. These are bacterial chlorophyll A molecules without the magnesium ion, and they're called bacteriophyllophytin, and shown here in blue. Eventually, bacteriophyllophytin will pass those electrons to the quinones, shown over here in light orange, and the quinones will be able then to pass those electrons to the next transport system within the electron transport chain. In bacteria, however, the electron transport chains are not Z-shaped as they are in cyanobacteria or plants, but they are actually circular. For example, when we take a look at a purple photosynthetic bacteria, we see that the electrons are being passed in a cyclical fashion. Here to the left, what we have is the redox potential of the components of the electron transport chain, and as you can see, we're going to begin over here with a chlorophyll molecule, a bacterial chlorophyll molecule, that absorbs light at the wavelength of 870, which is in the far red spectrum. Once that molecule absorbs light, it's going to go from being a really poor electron donor with a redox potential of 0.5 to being an excellent electron donor with a redox potential of -1 volts. Electrons from that chlorophyll will be passed to the bacterial chlorophyll, the second pair, and now from that pair to the bacterial phyophytin. Here you begin the passing of the electrons to the quinone pool, and the quinone pool eventually are going to bring it to the Cytochrome BC1 system. The Cytochrome BC1 system is able eventually to pass it to the Cytochrome C2, which is a carrier, which then is able to bring them back to the unexcited chlorophyll 870. As you can appreciate, the electrons are being passed directly through a cyclical movement. So this is called cyclic photophosphorylation. Now, electrons that are coming through the Cytochrome BC1 system are able then to fuel the pumping of protons across the membrane, which generates a proton motile force that can be used for making ATP. Now, eventually, the electrons being lost in the system are going to be taken from an inorganic molecule. Hydrogen sulfide, thiosulfate, sulfur, or ferrous irons are molecules that are able to be oxidized, donating the electrons to the system. So here we have a depiction of the system again. The energy harvested from light will be passed by the light harvesting antenna all the way down to the reaction center in the form of resonance. The bacterial chlorophyll in the reaction center, it's going to become a really good electron donor once it receives this energy, and it will pass those electrons to bacterial phyophytin, which eventually brings it to the quinone pool. Now the protons are going to be passed to the quinone pool and eventually will come to the Cytochrome BC1 system, which is able then to pass them around. Now from here, they go back to Cytochrome C2 and are donated back to the chlorophyll molecule. The protons that are being passed from the cytoplasm to the periplasm of the microorganism are then be able to be used to fuel the formation of ATP. So what you can see from this graph is that many different kinds of anoxygenic phototropes are using a cyclical photosystem in which one bacterial chlorophyll is going to pick up energy from light and go from being a really poor electron donor to an excellent electron donor. The other thing that I want you to see from here is the different wavelengths by which the bacterial chlorophyll are able to absorb light, and that not all the systems are having the same kind of electron transport molecules. Purple bacteria uses Cytochrome C2 versus the green sulfur and the heliobacteria using the Cytochrome C553. So as a difference from the photoautotropes, which include cyanobacteria, the electron flow in the anoxygenic bacteria is always cyclical, and the electrons are being released from the reaction center. They flow through the electron transport chain and return again to the reaction center. They are not going to be utilized to reduce an organic compound. Now, the last type of photosynthesis we'll look at is oxygenic photosynthesis. This will be somewhat of a review from the material that you learn in Bio 110. As you know, oxygenic photosynthesis can be divided into 4 stages: the absorption of light, where you're going to have a generation of high energy electrons, and the formation of oxygen from splitting water. You're going to have the electron transport leading to the production of NADPH and the subsequent generation of the proton motile force, the synthesis of ATP by the ATP synthase, as well as the conversion of CO2 into carbohydrates during carbon fixation. So take a look at the stages in photosynthesis. All of them are tightly coupled and controlled to make sure that the microorganism is going to produce the carbohydrates that it needs. And they are catalyzed by multi-protein complexes that are found in the membrane of the microorganism, or in the thylakoid membranes inside chloroplast. The part of the absorption of light, the production of NADPH, and synthesis of ATP are considered light reactions. The synthesis and fixation of carbon dioxide is considered the dark reaction, and that is going to happen in the cytoplasm of microorganisms or in the stroma of chloroplast. As the difference from anoxygenic photosynthesis, oxygenic photosynthesis happens in a Z scheme, where you have two different photosystems working together to generate sufficiently energized electron carriers to generate NADPH. What I'm going to do now is to walk you again through the oxygenic photosynthesis as it is shown in this image. Remember that this type of photosynthesis happens in plants, algae, as well as cyanobacteria. So let's take a look at the first photosystem, and that is photosystem 2. Photosystem 2, it's the place where water will be split to generate oxygen. And at that point, you will have light that is going to be harnessed and used to bring the chlorophyll 680 from being a very poor electron donor to making an excellent electron donor. Now, you're going to have a series of electron transfer steps that are eventually going to pass the electrons to the Cytochrome BF complex. And in the Cytochrome BF complex, you're going to generate the proton motile force that is going to help with ATP production. Now, the electrons from the Cytochrome BF system will eventually pass to phycocyanin, and from phycocyanin will arrive to the P700 system. That is the photosystem 1. Light again is going to be used now to get that P700 chlorophyll from being a poor electron donor to an excellent electron donor. And eventually, by a series of reactions, you're going to get the passing of electrons to the NADPH reductase, which is going to produce NADPH to generate the sugars that the cell needs. As shown in this image, you can pass electrons from ferredoxin, which is part of the photosystem 2, to the Cytochrome B6 system in a cyclical fashion, and that only happens when hydrogen sulfide is present. Hydrogen sulfide is able to inhibit photosystem 2 and now help with the generation of that cyclical system, just as in anoxygenic bacteria. Now, that is going to forsake the production of NADPH, but it's going to continue helping with the proton motile force to generate ATP. Now, autotrophic bacteria, once they have generated the ATP and NADPH they need, they will use those high energy electron and high energy carriers to make molecules through the Calvin cycle. So here in this image, we have some of the important reactions in the Calvin cycle that are going to help fix CO2. Let's begin by the fact that the Calvin cycle is going to utilize two important enzymes, the ribulose bisphosphate carboxylase or Rubisco, and the phosphoribokinase. Now, Rubisco, it's the most abundant enzyme that we know because it is used by plants and cyanobacteria to fix CO2. So a ribulose biphosphate is going to be condensed and carboxylated with a carbon dioxide to generate a unstable intermediate that eventually gets split into 2-phosphoglyceric acids, or PGA. Now, eventually those 2 PGA will be phosphorylated by some of the reverse steps in glycolysis to generate 1,3-biphosphoglyceric acid, and then that will be reduced to generate glyceraldehyde triphosphate. Glyceraldehyde triphosphate can eventually be used for generating molecules by biosynthesis in a reversal of the glycolytic system. Now, to reset the system, ribulose 5-phosphate will be phosphorylated by the phosphoribulose kinase to generate ribulose biphosphate, and that will then repeat the cycle. At the end, what you're going to require is 12 NADPHs and 18 ATPs in order to fix 6 carbon dioxide molecules. So the steps of carboxylation from 6 CO2 molecules and 6 ribulose 1,3-biphosphate, you're going to generate 12 3-phosphoglycerate. 12 ATPs are going to come into play in an energy input reaction to have 12 1,3-bisphosphoglycerates. Here, 12 NADPHs are going to donate electrons to reduce the 1,3-biphosphoglycerate into glyceraldehyde triphosphate. And eventually, you're going to remove a 6-carbon molecule for biosynthesis, generating, for example, a fructose, and there you're going to have 10 glyceraldehyde triphosphates, which by various different rearrangement reactions will become 6 ribulose 5-phosphate. Phosphoribullokinase will eventually phosphorylate those 6 ribulose 5-phosphates to generate the 6 ribulose 1,5-bisphosphates, and the cycle begins again. So overall, the stoichiometry of this reaction is going to take 6 carbon dioxides, 12 NADPH, 18 ATPs to generate a sugar with a phosphate, plus 12 NADP plus and 18 ADP and 17 phosphates. In some microorganisms, this is going to happen in inclusion structures called carboxysomes. These structures have a thin protein membrane and a crystalline array of rubisco inside of them, which is going to help it exclude rubisco away from oxygen. You also have another enzyme present called carbonic anhydrase, and that enzyme is allowing you to take carbonate and convert it into CO2 that eventually will fixate it. So this will conclude the photosynthesis lecture, and this will be the last lecture that we included on the exam on Thursday. Have a good day, and we'll see you tomorrow.